Okay, so good morning, everybody. Welcome now to our very last class. It's hard to believe that it's gone so quickly, but it has. And we're going to finish off by discussing the last part of our segment on plants. And we've already learned something about the kinds of tissues that occur in plant stems, etc. And we finished off last time by beginning to discuss the types of growth that can actually occur in plants. We know already plant growth is indeterminate and increases in length of the plant stems and of the roots occur from the apex, from the apices of the shoots and the apices of the roots. And that is what increases the length of the parts of those parts of the plant. But we also have to remember that plants actually increase in diameter as well, and they increase in diameter as they grow. They also increase in diameter as they age, and that is going to be the subject of a lot of our discussion today. Is how is it that they actually manage to increase in diameter? We understand quite well now how they increase in length. The answer is that the initial meristems, the initial dividing cell groups that are produced in the plant are called the primary meristems, and those are the ones that lead to the initial structure of the stem. And we saw the distribution of the different kinds of tissue, the ground tissue, and then the vascular tissues. But those, as the plant ages, in fact, as we look at a plant stem, if we go down the plant stem, the older parts of the plant stem, the primary meristem gives way to new kinds of meristem. And these are called the lateral meristems, and they are what is responsible for causing the increase in diameter of the plant stem. So the lateral meristems that we're going to be concerned with are these. First of all, there is a circle of meristem called the Vascular Cambium. Now, this is in eudicots plants. Okay, this is not in monocot plants. Those I'll mention a bit later. It's a little bit different. So what you can picture is here is this circle of meristematic tissue. We call it the cambium. It's called the vascular cambium in this case. And the outside here, there is another cambium, and it's called the Cork Cambium. So what is going to happen is that these meristems are actually going to effectively displace the initial tissues that were present. And they do it by division. So if you have a look at the vascular cambium, these red cells here, they divide and produce cells on either side. They produce on the inside here, they produce xylem. And on the outside, they produce phloem cells. These xylem cells are persistent. That is, that they are so heavily thickened, the cells, that they persist from year to year. The phloem cells, on the other hand, are fragile, and they tend to be crushed. So you can think of this like, almost like a conveyor belt of cells being pushed out. At the same time, these cambiums here, they can increase in diameter because they are able to divide sideways as well and grow. The cells, once they've divided, they increase in size. So those cambiums increase in diameter and continually produce cells to the inside on the vascular cambium and to the outside of the vascular cambium as well. The Cork cambium only produces cells to the outside. And that is the tissue it produces is cork, which replaces the epidermis. The epidermis usually just dies and sloughs off. In some trees, it persists, but in most, the epidermis dies and is sloughed off and is replaced by the thick layer of cork. That layer of cork is important. We'll talk about its functions in a minute. Okay, so if we were to look at any plant, what we can see is that every year it undergoes a growth spurt and will produce a length of stem with internodes, nodes, etc., etc. There's usually only on relatively young stem that leaves are born. And if a tree is a deciduous tree, if it drops its leaves in winter, then we can see the leaf scars that are left behind. A new stem, of course, ready to grow in the spring from the apical bud there. So in the axes of the leaves, there are small areas of meristem, which are called the axillary buds. And those have the potential to grow into new leaves, flowers, or into thorns, in actual fact. They can also produce the side shoots. This side shoot will arise from an axillary bud there. So when we look at plants, you probably know this already. Some plants complete their entire life cycle within a season, within 1 year. And those we call the annuals. The annuals usually drop seed, which is dormant over the winter, and will then germinate in the spring to produce a new generation of adult plants. Many plants have a 2-year life cycle. They're called biennials. And what usually happens with the biennials is that they spend 1 season building the plant effectively, building up storage. They persist over a winter season. And in the second season, they produce flowers and seed. And then the plant, the parent plant, dies. Biennials are very important to us. Many of our food crops, in actual fact, are biennials. I'll give you a good example. That is onions. Onions actually have a 2-year life cycle. If you grow onions, you know that in your first year, you get the onion which we use. If you leave onions in the ground, you leave them the next year, that bulb, which is actually a collection of leaves, of thickened leaves filled with storage material, in the second year, that will sprout a flower, and then the whole plant dies. So things like onions are biennials. But there are many plants which are extremely long-lived, which live from year to year to year to year. All the trees, for example, all those kinds of plants. Many, many plants are long-lived, and we call those the perennials. Okay, so we've learned now that primary growth arises from the apical buds, and the plant grows by increasing its length. But then in some plants, they will continue to grow sideways and increase their diameter. This growth in length we refer to as primary growth. In this increase in diameter, we call secondary growth, and we're going to spend a little time talking about how secondary growth occurs and what its impact on anatomy is. But there are plants where most of the growth is simply primary growth. There's very little or no secondary growth, and those we call the herbaceous plants. And many of the annual plants, for example, do not undergo any secondary growth at all. But it's in those plants which do, the characteristic of them, of the plants that undergo extensive secondary growth, is that they produce a tissue which we call wood. Wood is the result of processes that occur during secondary growth. And in something like a tree, for example, both processes are going on at the same time. You've got primary growth going on at the tips of the branches, and you've got extensive secondary growth occurring further down the plant. Okay, so have a look at this actual anatomy. We know something about the different kinds of tissue, but let's have a look at the actual structure and anatomy of the stems. The young stems are usually covered by an epidermis, and that epidermis is covered with a cuticle. that is cellulose and waxy materials, which effectively seal the tissue and limit water loss from the tissues. Then on the body of the plant initially is built of ground tissue, cortex outside the vascular tissue and pith inside. In the monocotyledons, there's no such distinction, as we'll see in a minute. In the monocotyledons, it's all a ground tissue. It's not referred to as pith or cortex. And the ground tissue is, its basic form is this kind of cell. And these are parenchyma cells. And they are thin-walled cells. And in this case, there is here no secondary thickening. We refer to this as being the primary cell wall, and it is composed only of cellulose. And you can see it's just, it appears like a thin line. If you look very closely, you can actually see where the cell walls of two cells abut on one another. But there's no other thickening taking place in this tissue. But this parenchyma can be thickened, and when it is thickened, we call it turns into other kinds of tissues. And the most important ones are called collenchyma and sclerenchyma. And there are here, for example, this is just ordinary parenchyma tissue. A parenchyma tissue is often characterized by having these little gaps between the cells. You see there. And these are important because they are channels for passage of gas. And of course, these cells are respiring and they're live. So they're metabolizing. So they need oxygen, and they need to void their carbon dioxide, and it moves through these channels. Collenchyma cells have been thickened. They have been secondarily thickened. Here you see this thin line here. That's the original, the primary cell wall. This material here is lignin, a very, very, very tough, strong, long-lived polysaccharide. And it's laid down around the primary cell walls. Collenchyma, characteristically, it occurs it's thin in some parts and thick in others. It's thick in the corners, thin at the sides, so it's unevenly distributed. There's another kind of tissue called sclerenchyma. And sclerenchyma is tissue which has been very heavily thickened, and the thickness is uniform. And you can see here that there is in the center, there is a lumen, the central part of the cell, and it's surrounded by this very dense secondary thickening. Sclerenchyma can occur in various parts of the plant, but we very often see a little cap of sclerenchyma above the vascular bundles. And we'll probably point that out later, if I remember. Okay, so yeah, there are some other strange kinds of sclerenchyma. When sclerenchyma becomes extremely heavily thickened, we call it scleride. This here, though, is one I want to point out to you. This is called a stone cell. And this is a scleride cell. It's very irregularly shaped, very heavily thickened. And I'm sure you've all had experience of sclerides, because I'll give you one good example. They occur in a fruit like pears, for example. You know, you can eat a pear, and pears have that pleasant, crunchy kind of texture to them, as well as the soft, sweet fruit. You have this crunchy bits. Those are sclerides. And they are supportive. All of these tissues here are supportive tissues, and also may provide some protection as well, because they are tough. Okay, so the really important thing to say about the stem anatomy is that the organization of stems in the eudicotyledons and in the monocotyledons is quite different, and it has important implications. So first of all, this is a monocotyledonous stem. Monocotyledonous plants do not have those secondary meristems that we've talked about before. They do not have vascular cambium and cork cambium. Instead, they only have little patches of procambium, which each produce a vascular bundle, and they are irregularly scattered through the stem. The stem can increase in diameter, but it increases in diameter because it adds ground tissue. That's all. So even in an old monocotyledonous stem, you cut it across and you look at the living tissue, and you'll see that there are all of these individual vascular bundles scattered throughout this ground tissue. In the eudicotyledonous plants, however, the organization is completely different. If you look at a young eudicotyledonous stem, you'll see in the center, here is the pith and of parenchyma cells. But there is around here an organized cylinder of the vascular tissue with phloem to the outside and xylem to the inside. And this arrangement here allows the plant to lay down year after year after year, to lay down layers of xylem to the inside and phloem to the outside. This, if we look into very young stem, we're looking at the procambium that produces individual vascular bundles. But as the stem ages, as we look at older and older stem, what we find is that primary meristem is replaced by vascular cambium, and the vascular cambium extends between these vascular bundles to form a complete cylinder, and it begins to lay down cylinders of xylem to the inside and cylinders of phloem to the outside. So the anatomy of the stem changes. Now, in the monocotyledons, that can't happen, okay, because they just have their vascular bundles scattered throughout the stem. They can't lay down these concentric rings of growth. And that has an important implication. It means that monocotyledonous plants are extremely limited in the amount of, in the length, for example, of the stem, because they cannot build up the same strength in the stem that the eudicotyledonous plants can. Cylinders of material are extremely, extremely strong. And so the stem of a eudicotyledonous plant is a very powerful thing. Its anatomy allows it to become very tall. That's how the giant sequoias can be hundreds of feet tall. Remembering that they grow every year. Even the very old plants are still growing at their apex. But the body of the plant can be supported by the rings of this very strong tissue that is going to be laid down, as we'll see in a minute. Monocotyledonous plants can't do that. They can't lay down these rings, and they are very limited in height. What monocotyledonous plants do, if those that are tall, because there are some which are quite tall. The palm trees, for example, you can think about the palm trees. Well, they don't reach the height of sequoias and things like that. But the monocotyledonous plants, in order to build up stem strength, what they do is they allow their leaves to grow very close to the stem. As they come out, they grow very close to the stem with a very thick, fibrous portion to the bottom. And then when the leaf dies and the leaf falls off, they leave behind those leaf bases, and those leaf bases encircle the stem, and provide the strength which allow them to to grow to a certain extent, to become quite high. But they are very limited compared to eudicots and to coniferous plants. Coniferous plants have a somewhat similar kind of growth to the eudicots. Okay, so let's have a look now at the anatomy of the really important part of the plant, which are the leaves. The stem grows to a height for a specific purpose, and that is so that it can lift its leaves as high as possible. And usually what plants are attempting to do is to lift their leaves into an optimal position so that they can photosynthesize effectively. But it's the leaves that are the major photosynthetic organs for most plants, not all, but for most plants, the leaves are the major photosynthetic organ. I say for most because you need to think, remember, cacti, for example, have no leaves, right? Well, they do have leaves, but they kind of disappear very early on. And what constitutes the body of the cactus is in fact the stem, which is the major photosynthetic organ. So there are specializations, but in most plants, it is the leaves that are the major photosynthetic organ. So the leaves develop from specialized meristematic tissue, which is produced which specializes right behind the apex, right behind the apical meristem. Here's a shoot, and here's the shoot apical meristem that is increasing the length of the plant. But right behind it here, there is this meristematic tissue here, which is rapidly dividing and will soon start to specialize and produce the leaf anatomy. They don't look like leaves at all. They're the nascent leaf, and we call them the leaf primordia. At the same time as the leaf primordia are developing, a small area of meristematic tissue is preserved right there in the axis there. And if we go a little bit further back, here is a developing leaf, and here's the stem, and in that axis there, here's a little bundle of meristem, and that's the axillary bud. That is how the axillary bud is formed. Okay, so first of all, the leaf structure is all designed as a photosynthesis machine. And the leaf is held in a particular attitude towards the light, and its strength is given to it by the vascular structure, the vascular system which runs through it. The leaf is thin. It is thin usually for a reason, and the reason is that there must be an efficient exchange of gas inside the leaf amongst the cells that comprise the leaf, because that's where photosynthesis is taking place. And photosynthesis needs carbon dioxide as a base product. It needs water, which is being supplied by the plant, but is also going to release oxygen, and oxygen has to be voided from the leaf. If it's not voided, if it builds up too high, it starts to cause real problems with photosynthesis. So there has to be a flow of oxygen and carbon dioxide through the leaf. At the same time, it's very easy for the cells inside the leaf to begin to lose water by evaporation. It begins to pass out of the leaf. That loss of water from the leaves we call transpiration. And it is one of the things which actually lifts fluids through the plant. But it can be very dangerous. And so it has to be carefully controlled. The leaves are usually covered. First of all, they have an epidermis. The epidermis of leaves is usually covered with a layer of cellulose and wax that forms a cuticle around the outside of the leaf. And that is a sealant. But then we have to allow gas exchange into the leaf. So there are specialized cells which form what we call the stomata. They are pores in the epidermis which allow gas exchange in and out. And those stomata are very carefully control the size of the pore. The hole this is what they look like. There's a hole here leading from the outside to the inside of the leaf. And this hole is called the stoma. On either side, there are cells, very specialized cells called the guard cells. And guard cells can open and close the stoma. And they will close the stoma when the plant is water stressed. They will only open when there's enough water, the plant is well watered. And by doing so, they ensure that when there's water stress, you don't have loss of water from the leaf that the plant cannot afford. Okay, so here's the leaf structure. It's actually quite complex, but we'll just look at it in simple terms. First of all, the structure of the leaf is preserved. It's the openness and the flatness and the attitude of the leaf towards the light and everything else is controlled by the vasculature, by the structure of the vasculature. In eudicots, it's usually stellate or a net system of veins running through the leaf. In the monocots, the veins are parallel, but they perform the same function. So they are providing structural support, but they're also, of course, conducting materials back and forth. And very importantly, they are conducting products of photosynthesis to the rest of the plant. Here is the main midrib of the vein. First of all, on the inside, it's like a big vascular bundle. So here is xylem there and phloem here. And around the vein of the leaf, there is a lining of cells called the bundle sheath cells. I can't go into all of the details about it, but the bundle sheath cells can be extremely, extremely important in the processing of product of photosynthesis. They're not just there to kind of delineate the vein. They actually have a very, very important physiological role to play, but we can't get into it. Above the veins, there's often a layer of sclerenchyma fibers, and that's why we can see the vein so easily, is because it's actually it's thickened with a layer of sclerenchyma above, which is protective of this delicate part of the vein, but also provides strength so that the structure of the leaf is preserved in three-dimensional space. The tissues of the leaf consist of a layer. First of all, let's just have a look. Here's the epidermis with a cuticle above it, and it's penetrated by stomata. Here's one here. The vast majority of the stomata lie on the underside of the leaf, but the top of the leaf can have some as well. But it's this tissue here which must be carefully aerated. Must have airflow going around it, so that they tend to be more predominant on the underneath of the leaf. So there's the epidermis there. This layer here, or this tissue, all of its tissue inside is called mesophyll, and it is photosynthetic. These are the tissues which have a gazillion chloroplasts in them, and they are the photosynthetic tissues. On the top of the leaf, they are organized into these long cells. You can see them in a section here. They have big vacuole, in a central vacuole, and then thin layer of cytoplasm and many, many, many chloroplasts. And these are organized so the cells all lie like that next to one another, and it's called the palisade mesophyll. But below that here, there is the cells which are very loosely organized and with lots of space in between. And that is called the spongy mesophyll. The leaf is thin enough that light can penetrate, well, it can actually penetrate right the way through. But as it passes through, the useful wavelengths of light are absorbed by the chloroplasts in both the spongy and the palisade mesophyll. So why is the leaf green, by the way? Do you know? Well, what it gives something its color. It's the wavelength of light which is reflected from the.
Is it the chloroplast?
Yes, but why are chloroplasts green?
Why are they green? The chlorophyll?
Yes, but yeah, you're right. But what I'm getting at is, if they are green, green light is being reflected into our eyes. The plant is not using green light, right? The plant is actually using blue and red light. It's blue and red light that are absorbed. It's the blue and red wavelengths that are absorbed by the chloroplasts and used to drive photosynthesis. Green is not used. Green is reflected from, and that's why we see it as green. We see the least useful wavelengths, which are just reflected. The rest are absorbed by the chlorophyll.
Okay, thank you.
So, okay, so as I pointed out to you, because of the organization of the stem structure, secondary growth, extensive secondary growth is limited to the eudicotyledonous plants and to the gymnosperms. The monocotyledonous plants, it's very difficult for them to undergo any kind of secondary growth. But when in a eudicotyledonous plant, we'll have a look here and realize that when we're looking at a branch or at the stem itself, what we're remembering is that they have indeterminate growth. There's only one part of a plant which is usually determinate in its growth, and that's the leaf. Leaves, in most cases, leaves have a determinate size to which they will grow, and then they stop. But the stems and the roots, growth is indeterminate. It carries on throughout the lifespan of the plant. So if even in a very old tree, if we go to the young shoots, what we're going to see there is primary growth adding length to the stem. And if we cut across a young stem like that, what we'll see is that typical primary organization. And that is that there's an epidermis to the outside. There is cortical ground tissue there, pith in the middle, and then the vascular bundles are organized as discrete bundles like this. And they have arisen originally from what is called the procambium, the primary meristem, primary vascular meristem that has produced these initial bundles. However, very soon after that, in the same year and in the same growth spurt, as we go down the stem and we look as it begins to age, we find that this original meristem here is replaced, and it is replaced by vascular cambium. The vascular cambium has the characteristic, it's not limited to the vascular bundles the way the procambium was. Instead, you can see here, it extends between the vascular bundles, and it now has the potential to produce cylinders of tissue rather than these discrete bundles. There's vascular cambium there, but here is the result of that vascular cambium beginning to produce secondary growth. Here's secondary xylem to the inside, secondary phloem to the outside, and you can still see the remains of the original vascular bundles there, but they are being replaced now by these cylinders of vascular tissue. At the same time, on the outside, the meristems that produce the epidermis are replaced. The epidermis, as the stem begins to increase in size, usually, sometimes in some plants it's preserved, but in most plants, the epidermis is lost. It dies and sloughs off. And instead, a cambium, a meristem called the Cork Cambium develops. Here it is here. And it produces tissue only in one direction, and that is towards the outside. And it produces this thick layer. We now refer to this as being the periderm. And it's the outside layer of corky tissue. And cork is a specialized tissue. It is non-living, and it is thickened with a special substance called suberin, which is waterproof. So this bark to the outside performs two functions, and that is, it is protective of the structures of the stem, but it's also very waterproof, so that it stops the stem from losing water to the outside. The stem still needs some gas exchange. So there are special pores in the bark called the lenticels that allow some gas exchange to take place, but it is a controlled gas exchange. Okay, so as we get older and older, we find that this vascular cambium lays down rings of material. The tissue that it produces towards the inside is xylem tissue. And the xylem tissue is heavily thickened, and it will persist even when the cells have died. And that is what is going to produce what we refer to as being wood on the inside. On the outside, it's producing phloem. And as it produces phloem, it begins to crush the old phloem. It crushes the old phloem up against the cork there. And that the phloem, as a secondary phloem, as it's produced year by year, it actually doesn't, the old phloem doesn't persist. It gets crushed into here and is lost as a thin layer of dead material against the cork. The Cork Cambium has a very strange property, and that is it can die off and reform. So this is what happens. This layer of phloem here gets crushed into the cork there, and the Cork Cambium reforms on the inside here and begins to form new cork. So when we look at the outer layers of an old plant, what we're looking at is old crushed phloem and cork all sandwiched in together with one another. So let me just check that I've got everything. Oh yes, and the one thing I must mention, and that is that the ground tissues begin to disappear. It's difficult to find them. Some of them are still there. That's how, for example, the Cork Cambium can reform. It reforms from ground tissues that turn meristematic. But the pith on the center here begins to get crushed. And it will ultimately, when we look at a really old stem, we won't be able to see any pith at all. Instead, what we'll see is a big, thick layer of secondary xylem, and then a very thin layer of living phloem tissue, and a quite thick layer of the cork on the outside. And you know, you can, when you go to a tree, you can actually pull the bark off. Well, if you do that, what you're doing is you're stripping off all. What you're looking at is when you strip a piece of bark off on the very inside, what you're looking at is this cambium tissue here, the living phloem, and then the cork on the outside. That's why if you ring bark a tree, if you go and you strip the bark off a tree in a complete circle, the tree dies. And the reason it dies is that you have taken away the phloem tissue, and the flow of material, of photosynthetic nutrients to the roots stops, and the roots die. That's why ring barking a tree kills the tree. Let me check that I've covered everything. Okay, so yes, I have. Okay, so this is typical. This is a typical section of a piece of wood. And one of the things that we notice immediately, there's a distinct band structure to it. And here we see 1, 2, 3 of those bands. But look carefully inside the band, and you'll see there is actually another band, and that's of these smaller vessels and tracheids here. And then this area, big area here with big wide tracheids and vessels. So this is xylem tissue, heavily secondarily thickened and still active. It's actively transporting water up through through the plant. And this thinner layer here is the xylem tissue that is laid down in winter. And then this thicker layer here is the xylem which is laid down in summer, when the plant is really quickly growing, actively growing. The growth, of course, slows down prodigiously during winter. So we have this thin band, just, you can think of it as like maintenance. The plant is almost dormant, but it's maintaining its functions. Then the great, tremendous growth spurt in spring and the summer to produce this band. Then it shuts down again in the winter. And as a result, when we look at wood, we have these rings. We have a ring there, and we can count the rings. We can count the number of rings, and it tells us how many seasons of growth the plant has enjoyed. This is now a plant which has undergone a couple of years of secondary growth. The pith is almost lost in the center here. In very old tree, the pith is lost entirely. These each here, there's a growth ring there, there's a growth ring there, there's a growth ring there. The one thing I haven't mentioned, and I'm not going to go into any detail about it, is that there are ways in which this plant can move materials horizontally as well, because it has to make sure that all of the cells, the living cells, are supplied with nutrients, etc., etc. And that movement, horizontal movement, is done through these vascular rays here. Okay, so this plant has still preserved the old vascular bundles, and that you can see here still. Those are the remains of the original vascular bundles, and but then they are being lost. You can see here this has begun even here to form a cylinder of phloem tissue here. This will be eventually, the growth is become so extensive that this will be crushed into the cork. Here's the Cork Cambium there, and this is cork here. And if I was to strip the bark, this is what would come off. It would come off all the way from the vascular meristem there, the Vascular Cambium, all the way to there. That would be what constitutes the bark. The bark is not just the cork, it's all of this tissue as well. There's one other thing to point out, and that is that the phloem is renewed year by year. The xylem persists from year to year, but the xylem on towards the inside begins to lose function. It's the cells, most of these cells are dead anyway. The transport through xylem is largely passive transport, but the cells, the xylem towards the interior here loses its capacity to transport fluid. It just fills in and becomes essentially solid. So this is when we look at an old log cut across, for example. This is what we would see. this is the interior here. This wood is no longer transporting materials. It is dead. It is no longer transporting, no longer functional, and we call that the heart wood. Very, very tough and very, very strong. The layers around here, and it may be several layers. It's not that, you know, only this year's layer is like this. There may be several years of layers of xylem, which are still functional and which are still transporting fluids. And if we cut across a branch like that, and we look, we will see the plant begin to bleed through these vessels and everything of the xylem here. And that is called the sap wood. So sap wood is still actively transporting. Then we'd have the vascular cambium here, and that is what produces the xylem year by year. And then on the outside, there's secondary phloem that's produced each year, and the old phloem is crushed up against the cork in the periderm here. So we've got periderm there. Here's the phloem cylinder. Here's the vascular cambium. This is all xylem. And in this one, it's old enough that the pith has been completely lost. Okay, I'm just checking that I've covered everything. Okay, so this is in the eudicotyledonous plants. This organization of the vascular cambium and the production of the secondary growth is what allows these organisms to be so huge. And in fact, the largest land organisms are the trees like this. And in addition, we need to remember that the oldest organisms on land are also plants. And it's also thanks to this incredibly efficient organization of their anatomy and of their vasculature. And so there is, I know there's a question in your assignment says if you were to drive through this tunnel, what tissues would you pass through? And the answer is, of course, is big, thick periderm there, cork. And if you were to look carefully, you probably find the remains of ancient phloem crushed into it. Then you'd pass the vascular cambium. And then you'd pass through, first of all, the sap wood of the tree, then the harsh wood, and then back out the other way. Okay, so that Cork cambium is very important because it is what seals the trunk seals the stem of the plant above. When it's above ground, it is very susceptible to desiccation, to drying out, and it is the presence of the cork around it which enables the stem to resist the drying out. And I've told you already, the bark consists of all of the tissues external to the vascular cambium, and the cork is penetrated by these pores called lenticels, and that allows for gas exchange to take place to the living cells on the inside of the stem, which is essential because they are still respiring. Even though they're not photosynthesizing, they are respiring, and they are producing carbon dioxide, which has to be voided in some way. Okay, so let me just see. Yes, all right. So this is when you're doing your revision, this is a very good diagram to come and look at, because if you can explain what's happening here in this diagram, you'll explain everything about the nature of secondary growth. And just to remind you, here on the inside here, these are the remains of the original vascular bundles. That was the primary growth that produced those. And the cambium that produced it was called the procambium. The procambium is replaced by the vascular cambium, which extends in a ring. And so it is between the original vascular bundles, and it begins to produce these cylinders of growth. Here's the old phloem being crushed up against the cork, And this whole layer here, produced by the Cork Cambium, is called the periderm. And eventually, in fact, the Cork Cambium will reform on the inside here and crush the old phloem into a new layer, into new layers of the cork. That is not an annual, you don't see kind of annual rings in that. That's, there are other things which govern when the Cork Cambium is reformed. But the net effect is that for both from the growth of the periderm and the adding of vascular tissues on the inside of the cell, that is what is going to increase the diameter of a stem. And that is, or by definition, the secondary growth. This is, I think, the last slide. And oops, sorry. And again, it is useful for in terms of your revision. I'm sorry. Here we go. Oh, just leave it. Yeah, this is going to be useful for you in terms of your revision, because it will shows you as we proceed back, like from the youngest part of the stem downwards, it will show you exactly what the fate of the different meristems and of what they produce. And so, for example, the ground initially, the ground meristem produces the cork and the cortex and the pith. But this ground meristem is interesting because it retains the ability to form new meristems. And so, for example, the Cork Cambium forms actually from ground tissues. That's how it can form and then reform. Even in those highly organized secondarily thickened stems, where we seem to see only xylem and phloem and everything. Mixed in with that tissue, there is still some ground tissue. It's just not organized the way it was before, but there are still ground tissue cells mixed throughout the other tissues. The first cambium, the first meristem, is called the procambium, and that is what produces the initial vascular bundles, the primary phloem and the secondary phloem. But that is replaced by vascular cambium, and the vascular cambium is what produces secondary phloem and secondary xylem. The protoderm is the early meristem that produces the epidermis. And unless we're looking at an annual without any secondary thickening, the protoderm is lost and is replaced by the Cork Cambium, which produces the cork, and the epidermis sloughs off. So this basically gives you a summary of the origin and the fate of these different tissues. Okay, so we leave it there. Does anybody have any questions?
I just have a quick question. So we do have class on Friday, right? That will be like our last class.
No. Today is your last class.
Today?
Yes. Semester ends on today, the 5th of May. Originally your exam was set for Friday, but I've moved your exam to 1 week from today. So your exam will be on next Monday. But we are done. Are you devastated?
I am, because I'm still thinking that I have. I'm like, yeah, I got 1 more week before finals. Okay.
No.
Wow.
No. You need to go and check your calendar. Okay.
Okay.
All right.
Well, it was nice being in this class. I hope to see you on campus.
Oh yes, it will be very nice actually to be able to actually see people and on campus again. And yes, I hope. I'm not sure what your classes are going to be, but if you see me wandering around, do come up and tell me, so that we can say hello properly.
Will do. And then I think you also teach genetics.
No, I'm not teaching genetics. I'm teaching biology again, the first semester, and I'm teaching forensic science and human biology, I think. Those
Okay,
are
so
what
when I get to those courses, I'll be sure to take your class again.
All right. I'll be very happy. All right.
Okay.
Good luck, everybody, with as you finish your revision and everything. But everybody's done so well in tests so far. I'm sure that you're not going to have any problem.
I just wanted to make sure this coming Monday, the 10th, is the final.
That is correct. Yes.
Okay.
I moved it to the Monday.
Okay. Thank you.
I'll send out another notice just to remind people.
Okay. Thank you. All right. Enjoy your summer.
You too.